Analysis Center for one of NCI's big projects, or was, and Thomas led our work on thymoma, which is pretty awesome. It's in his thesis work. And then I think as part of working on these projects, I think it's a good example that that he got this pretty cool idea on how to mine it to find possible vulnerabilities in tumor samples. Yeah. And some of the results are almost too good to be true. And so we're now thinking about, like, can we actually take it into the lab? And so as folks listen to this, maybe have your thinking caps on, like what could be a cool next step to actually translate this into the clinic. But anyway, it's got a lot of potential, for very near future stuff. So anyway, I'll turn it over to you, Thomas.
Thanks, yeah. Thanks, Josh, for the intro. So yeah, today I'm just going to go over the work that I've done in the Stuart Lab. It's just really 5 sections. The first section, which Josh already talked about, was my work in thymoma as part of the cancer genome atlas. And then I'll talk about the sort of problem statement that kind of motivates my research, and then a description of the method, and then some results from that method, and then some future directions. So first off, this thymoma group, I was part of the thymoma analysis working group, which is part of the Cancer Genome Atlas Consortia. My job, this is really the first big project that I was exposed to, and here I used a couple of well-known Stuart Lab tools, Paradigm and Tumor Map, which we'll talk a little bit about later. The clinicians on this wanted to find links between histology and the molecular landscape of thymoma. And this culminated in the largest study of thymoma and thymic epithelial tumors to date. So, pretty cool. So to start, thymoma is a cancer of the thymus, both the epithelium around the thymus as well as the inside of the thymus itself. The thymus is between your lungs, and it functions in the immune system as a place where T cells mature. There's 2 different flavors of thymoma. A few of these are more malignant than others, but they're all histologically defined. Basically, you look at them under a microscope and they will look slightly different, and they're just put into these groups. If you haven't heard of thymoma, that's because there's only 0.15 cases in 100,000 patient years, so moderately rare. If you do get thymoma, there's a 69% survival rate at 5 years. That's overall survival, which compared to other cancers is actually encouraging. Some of the risk factors of having it, given that the thymus is part of the immune system, there's autoimmune diseases that come along with that. A third of all patients are going to have autoimmune disorders, whether it's RA or Sjögren's. The most common, though, is myasthenia gravis, which attacks the connective tissue. In fact, 75% of all MG patients have some sort of malignancy of the thymus. The treatment is surgical resection of the thymus. You can live without it, but you'll have autoimmune type problems later in life. But yeah, that's the background. As far as relating to other cancers, we did this mutational analysis. We compared all of these different types of cancers. And besides these 2, these are 2 childhood cancers. Thymoma has the lowest mutational burden. The highest one would be melanoma, and the next is small cell lung cancer. So even though it's low mutations overall, there are some that kind of stood out in our analysis. 3 that stood out, HRAS, NRAS, and TP53. These were found in the more aggressive forms of thymoma over here. This is the type B3 and thymic carcinomas. But then we found this other one called GTF2I. This is only mutated in thymoma. No other patients in TCGA have a GTF2I mutation. So this is a unique marker for thymoma, which is pretty cool. But going beyond mutation, this analysis working group had to factor in a bunch of different streams of data. We had rPPA, reverse protein phase array, copy number, mRNA, microRNA, and DNA methylation. And we were able to run 2 different methods. First, tumor map, which I'll explain a little bit, and Paradigm. And both of these came out of the Stuart Lab. And I can't talk about Paradigm without showing the Paradigm slide. This was work that Charles Vasky worked on in this lab, where basically you can take two streams of data, RNA-seq data and copy number data. You bring in a protein interaction network up here, and you put all those together and you can find inferred pathway levels, inferred molecular pathway levels for different samples. And we'll just refer to these as IPAs. The next method was tumor map. This was the work largely done by Julia Newton. Basically what tumor map does is it takes a bunch of different samples and different data for those samples, and then it kind of relates them together. It sort of clusters them by similarity. And there are some details here, but the net is that you get this map that looks like this. And it's actually a map. It's built on Google Maps API. It's this sort of top-down view, like an archipelago, of all of the different samples. And all the ones that are the most related get stuck together in these islands. So we wanted to run this on the Thymoma dataset. And we did a variety of different streams of data into the tumor map. We used RPPA, we used copy number data, expression data, we used the paradigm outputs, and we used DNA methylation data. We took all of those, we combined them, transformed them into the same space based on z-scores, averaged those, and came up with a single sample by sample matrix, and ran that into tumor map, and these are the results. So this is the figure 3 from the Thymoma paper. What you see here are each one of these little dots is a patient, and they're colored by different subtypes, different histological subtypes. So A is in the dark red, A, B is the pink one, the Bs are the sort of light blues, and thymic carcinoma is this dark blue one. And they all cluster fairly cleanly. So the silhouette score was something like 0.7 on this. The A-like cluster up here is almost exclusively A with some ABs mixed in. But these are known for having G2F2I mutations, certain overexpression, and then all of these underneath are the different inferred pathway level activities with just simple up or down arrows saying if they are active or inactive for those samples. And what we see is actually pretty encouraging. All of the samples roughly segregate with their histological types. So the B-like cluster is way off on its own. The C-like cluster is very far off on its own. In fact, the only one that's not part of the C-like cluster is up here, this dark blue one, the single dark blue one, which could be a lot of reasons for that, but these are all colored based on histology. And with histology, there's always some subjectivity with it. But the clinicians really liked this. They actually really like tumor map a lot. I did a face-to-face in Indiana, and I was able to do a live demo of tumor map just from UCSC's MedBook website, and I had clinicians come up and actually change attributes and recolor this graph, and they really liked it because it was super intuitive. And this ultimately culminated in this, this work published last year, February. The largest thymoma study to date. So I think it was 117 patients total. So the reason why I talked about that first is one, that's the first thing that I worked on. Two, a lot of the work that I did was was largely based on differential analysis. That differential analysis was, I thought, pretty interesting, and I kind of wanted to apply that to more cancer research, kind of beyond thymoma. So today I'm going to give you here just a quick background about the problem that I'm trying to solve and a few of the issues kind of surrounding our search for new targetable therapies in cancer. So, I mean, everyone in this room knows this, cancer is still an unmet need. 2005, it was responsible for 1 in 7 deaths worldwide. Now it's 1 in 6, or this was in 2015, but 1 in 6. The number of new global cancer cases is going to almost double by 2030, and the cost is also going to almost double. This number up here, $458 billion, that's in direct healthcare spending, but when you factor in the total economic burden of cancer, It's closer to $1.5 trillion by 2030. So it's pretty serious. This affects everybody, whether they have cancer or not. One of the direct costs of cancer is the cost of the drugs themselves. There's two main classes of drugs. There are large molecule biologics. These are the things you go into an infusion center for. And then small molecules, which you can actually take orally. In 2017, biologics, of the total amount spent on drugs, were about 1/3. So 1/3 of all the spending was on biologics. However, biologics only comprise 2% of all prescribed therapies. So clearly there is a huge cost difference between biologics and small molecules. And just based on the clinical trial schemes between these two, and based on the R&D expense and the manufacturing expense and the logistic expense, it's clear why biologics would be more expensive. So I'm thinking, there's a little bit of tapering off for small molecules. I think they deliver a lot of value to both patients and to payers. So why don't we find some targets for small molecules? Now, one of the issues with small molecules is the off-target effects, right? You think of chemotherapy, and like you already just think about all the different issues with chemotherapy, right? The hair loss, the nausea, everything that comes with it. That's because traditional oral chemotherapies have a narrow therapeutic window. What that means is, I wish I had my stick.
Yeah, right here.
Boom. Thank you very much. What you have here is a narrow therapeutic response. The green curve is the desired effect. The blue one is the adverse effect. And it doesn't take that much change in dose for it to go from something that's good to something that's bad. You want a wide therapeutic ratio, one where there's a big difference between desired and adverse effect. The question is, how do you find those? I think the modality to find those is through synthetic lethality. Now, synthetic lethality is this idea where, say you have two different genes, gene A and gene B. If both gene A and gene B are okay, meaning you have them, the cell's functional. If it loses A, that's okay, it can rely on B. Or if only B is missing and A is there, that's okay too, it can rely on A. But if they're both missing, that could lead to a synthetic lethal phenotype, meaning that the cell gets sick and the cell dies, which is a good thing, because we're talking about cancer, right? So the question is how do you find these synthetic lethals? Well, and how do they relate to cancer? These are the hallmarks of cancer from Hanahan and Weinberg. If we zoom into one of these hallmarks, it's genome instability. Genome instability is what's going to cause either that A or that B gene to be missing. So I think synthetic lethality, as far as a concept to find new druggable targets, is a valid one in cancer. This is actually a classic example of synthetic lethals that work in the clinic. Here is a normal cell, and there's two different genes involved with DNA repair, BRCA and PARP1. In a normal cell, if there's a break, it can rely on either BRCA or PARP1 to do the job of base excision repair. Great. In a cancer cell where BRCA is missing, it has to rely on PARP. Okay? If there is a drug that inhibits PARP that's given to a normal healthy cell, that's also okay too. The cell can rely on BRCA. However, if it's a cancer cell that's missing BRCA and has to rely on PARP, you give a PARP inhibitor, no repair will happen, a bunch of DNA breaks will accumulate in the cell, and it will ultimately cause cell death. This is the idea behind using PARP inhibitors in the clinic. And they're used in the clinic. These are actual examples of them. All of these, by the way, are in clinical trial now, except for this one. All these are in clinical trial for a mix of ovarian, small cell lung cancer, and I believe
Pancreatic recently.
pancreatic too, yes. I think it was this one, pancreatic. They're accurate. And I know some of you may be familiar with PARP, and know that there's some issues also behind this. There's some off-target effects to PARP, but the PARP inhibitor specific market is supposed to expand with a compound annual growth rate of 17%. This is just for PARPs, right? But clearly there's room in the market and desire for other synthetic lethals that are not PARPs. So I think we can find more of these targets. So I think synthetic lethality would be the way to begin doing this. Synthetic lethality was first that term was first coined by this Ukrainian geneticist named Dobzhansky, who did lots of fly experiments. This is Dobzhansky. This is his table of flies. All these little jars are different treatment groups, I'd imagine, for flies. I've never worked in a fly lab. I don't really know what's going on, but that's him. It started not with him. It actually started with another geneticist in 1922 doing similar work. He just came up with the name synthetic lethality. That was then. It kind of disappeared. The whole study of synthetic lethals kind of disappeared for a while. Came back strong in the 90s with yeast. All these yeast cells were screened in genomic screens, or in combinational knockout screens to find synthetic lethals. And now, in more modern times, like circa 2005 to around 2015, high throughput screening has been used to find synthetic lethal combinations of genes with both gene editing techniques like CRISPR and RNAi, as well as small molecules as well. But that's in a lab, physically, right, doing the work. Can we kind of guess? Can we make an educated guess? Or maybe infer some of these connections before we even try it physically? And there has been several attempts to do this. This one from Núria's lab in Barcelona, she used the synthetic lethals that were found in yeast cells and tried to map them to human cells. However, not all synthetic lethal network relationships are conserved between the two species. So, not the best comparison. This, Cyclops, was a good one. But this solely looked at hemizygous loss versus another gene's hemizygous loss. So it was just looking at copy number. I think we could bake in like other forms of data instead of just copy number. DAISY it came out in 2014, was patented actually the next year. And what DAISY does is it uses mutual exclusion, but it bases its data a lot on shRNA screens, which are inherently noisy. This was, I think, before they had Demeter, which was a method to take some of the noise out of these screens. So I think there's a lot of noise in this that is giving possible false positives for synthetic lethals, and it only looked at mutational context. SynLethDB is not a method to find it, it's just a compendium of different synthetic lethals. Select used mutual exclusion, but it only looked at copy number and mutation. That's it. So I think we can bake in a little bit more as far as different pieces of evidence to find these synthetic lethals, and I think the big one is expression, which all of these lack. So that brings me to the method that I'm talking about today, which is CLOVE. Stands for context loss dependent overexpression. And basically what it means is you have some missing gene, the context, which is gone, and you look for cases where the expression or overexpression is based on this context loss. I developed this in cancer cell lines, and I'll tell you the entire workflow. I used cell lines initially from CCLE. There's over 1,000 cell lines, 24 different tissue sites, a bunch of molecular data that's available for these. Screening data, because cell lines are used in actual physical screens, so there's that data. And for my previous work in target ID and validation, doing cell-based screening is a stepping stone to preclinical work for finding drugs and validating targets. Let's talk about input. There's two main classes of input for CLOVE. One is the context. The context are the genes that are reduced in each sample, like BRCA, BRCA is an example of that. Different versions that you could use is copy number or mutational calls from gistic or revealer. And it just looks like a matrix like this. Context, these are all the genes on the rows, and these are all the samples across the top, the columns. If you want to combine these two, you can. The way I do it is I just do an OR statement between all of the copy number alterations and all the mutation calls, and I come up with a combined context matrix. So you can do copy number, mutation, or both. The second piece of data that's an input is the compensation. These are gene expression in each sample. The example here is like PARP. You can take microarray data, which I use, RNA-seq data, which I've also used, into a similar looking matrix. Samples across the top, expression for all the rows. Expression, genes expression. Some pre-processing happens before you run clove. These are the two sample inputs that we talked about. The first thing that we do is we binarize this data. We say, I don't really care if something is like a little bit lost or very lost. I'm going to call it as either being there or not there. So I'm binarizing that data. You can use revealer to do this, and I do, or you can make some gistic cutoff. The next thing I do is I pre-filter. I take the expression, and I want to get rid of the confounding genes, the ones who are overexpressed, perhaps because of an amplification, or perhaps because of a subtype confounding factor, like basal versus luminal in breast cancer. I remove those. 2, when you load these, the user gets to pick what kind of filter parameters you pick. 2 of these graphs appear. They're distributions of the number of samples you have and the variance of those samples. You want to exclude things with a low variance and exclude things that have too few samples. The user gets to pick these, although there are defaults. When this data is loaded, these graphs will also appear, and these just tell you how many samples you can expect, or how many pairs you can expect to lose given the parameters that you pick. But there are defaults, so you don't have to do that if you don't want. The next step is to work on the null model. So this is the observed, right? This is after binarization and after filtering. This is expression after filtering. The null model is a permutation of the expression that effectively breaks the link between copy number and other context changes and the expression itself. So this gives you a randomized link between context and expression. You take both of those and you feed into clove. And you get some output which looks like this, context by expression, and you can form bipartite graphs from this as well. A little zoom up on what that computation is. It's this here. If it looks familiar, it's probably because it is. It's a Welch's t-test, where basically you're looking at the difference in mean from the expression when you are missing a gene versus the expression when you have a certain context gene. Positive cloves mean that you have an overexpression when you've lost a certain gene. You can think of this comparison kind of as a box plot. This is a single gene's expression. These are different context states. So this is when you lost a gene. This is when you have the gene. It's all good. The difference between these means is measured as a t-statistic, which is the clove. Right? You do that for the observed, and you do that for the permuted set, or the null model. But you don't want to do this just for a single gene pair, right? You want to do this for all of them. So what this is the Cartesian product of all of these genes in the expression input and all the genes in the context input, minus self-links. If BRCA is missing, it doesn't necessarily make sense to look at the expression of BRCA, because it's gone. So self-links are eliminated. When you compute all of the cloves, you'll generate 2 distributions like this, and you would expect to see, if it worked, this red curve shifted to the right. And I'll tell you why in a second. Now, all of these scores are based on a pair, right? A pair of expression and context genes. If you want to collapse those into a single vector, you can do that by just multiplying the CLOVE output and the context of a certain sample. This gives you the vulnerability vector for one sample for all of these genes. And essentially what this does is it counts the degree of a certain gene in a synthetic lethal network. This number, 2, for example, would mean that a certain gene has 2 different contexts that are pointing to it. So, what does the result look like? I ran CLOVE with 3 different contexts in breast cancer. Copy number, mutation, and that combined copy number and mutation for both the observed and the null in gray. And for all of these, I did a KS test between them. Copy number has a KS of 0.6. The next largest separation is mutation. And then the largest mutation of those 3 is the combination of copy number and mutation. And this is encouraging because the 3 observed distributions are shifted higher than the null distributions. And what that indicates is that on average, you see an increase of signal of an expression gain when you've lost a certain gene versus when you destroy that link. So CLOVE is showing some possible compensatory mechanisms or some possible expression-dependent changes. I repeated this process for all the different tissue types. The reason why I did tissue types is because I want to try to eliminate confounding factors comparing between different tissues. So I limited my search to just within tissues. This is the p-value of separation of observed from null. This is the t-statistic between those 2 distributions. The highest thing on the top right is cells from breast. They had the highest separation, or the most meaningful separation in CLOVES. And next were kidney, bone, and ganglia, and upper digestive tract. This line here is a p-value of 0.01, so anything above that is significant. But I focused on breast here because it had the biggest separation, or the biggest meaningful change. These are just medians of how these medians move from the null set to the observed set, and all of the observed ones are positive, and all of them are higher than null, which is encouraging. Then what I wanted to look at, this is in breast, what I wanted to look at were how CLOVE scored known synthetic lethals. So I computed a bunch of CLOVES in breast cancer, and I labeled them as being either seen in the SynLeth-DB database, a database of known synthetic lethals. These are ones that scored highly but were not known to be synthetic lethal. These are cis CLOVES, kind of interesting. The self-links that I was talking about, these are those. So it's expected that these would be a little bit lower than normal, and I can talk about something weird happening up here. And the nulls are just random. And there is a meaningful switch between these, or a meaningful boost up of CLOVE score in the knowns versus the potentials compared to null. The cis ones are interesting. The cis, what that means is you have a higher expression, if it's positive, means you have a higher expression when you've lost a copy of that gene. The topmost result in the hemizygous case, the topmost result was BOC2. BOC2 is known to be an indicator of aggressive ovarian cancer when it has a hemizygous loss, and is also needed in, it's also a high expression of BOC2 is needed to evade the immune system in a tumor. So kind of interesting up here. So CLOVE picks out knowns and gives them slightly higher scores. Great. The other way that I wanted to validate CLOVES was through something called congruency. Meaning if 2 genes are somewhat the same, they're going to have similar partners that they're linked up with in this synthetic lethal network. What I found was BRCA1 and BRCA2 had a very high relationship. They had a 0.35 Pearson coefficient when you looked at all of their different shared partners. If you did this outside of the module where these two genes belong, that correlation drops down to 0.073. So it's about 5 times higher between these two, which is interesting. The highest congruence was between these two genes. This was computed across all tissues. Was HRAS, proto-oncogene, and the serologically defined colon cancer antigen. These two genes had a similarity of 0.9 for a Pearson coefficient. And as it turns out, these are a known synthetic lethal. They were actually found in a RNAi screen in 2009, where these two were synthetically lethal with each other in both colon cancer and breast. So it's pretty encouraging as well. I wanted to take it a bit further and look at essentiality. So there's a bunch of essentiality scores out there. One compendium of this is DepMap. DepMap includes a variety of different kinds of essentiality scores, from shRNA to CRISPR to small molecule, and they have discrete calls. They make a call in a certain tissue, in a certain cell type, if a gene is essential or not essential. So what I did was I took all of my CLOVE scores, I took the vulnerability vector of them, that creates a single score of essentiality for a particular gene in a sample. I took all the cell lines that were intersected between my breast cancer analysis and their compendium. And what I found was the vulnerability vector, or the count or degree of how connected a certain gene is in a synthetic lethal network, is higher in every single of these 10 instances compared to the non-essential gene set. So it's showing that this vulnerability vectorization based on CLOVE scores is giving you higher scores that seem to associate well with essentiality. So again, one more piece of validation. Another striking one is in Demeter scores. These are also cell-based experimental data based on shRNA. Demeter does a couple of fancy things to get rid of some confounding factors in shRNA, like seed region bias. So that's why I chose this over the previous one, which is called Atari's. What I then did was I took, again, all of the cells that corresponded with my data and Demeter's data, and I ranked them and found the degree of concordance using this measure called Kendall's tau beta. And basically what it shows is that in the observed set, the correlation, each dot is a cell. Each of these cells has a higher correlation in the observed set when you compare it to its Demeter scores based on essentiality, based on the null, or compared to the null, which is almost 0. So this supports an association between vulnerability vectorization based on CLOVES and essentiality, or killing cancer cells, in an shRNA experiment in real life. Now, at the high level of CLOVES, meaning like highly scoring CLOVES, the ones that you would expect to be very essential, I was able to pick out this network. This is a network, this is a CLOVE network. So all these genes connected to each other show synthetic lethal relationships, all the black lines that you see. These are all implicated in apoptosis or avoiding apoptosis. These are all the context loss genes. These are all lost in a certain cancer cell, either due to mutation or due to copy number loss, and these are overexpression. So these are possible compensatory genes to all of these conditions. I just picked out a few of these that look familiar to me. For example,